I'm fine, hocam. I just got out of the exam. How are you?
Ah, how was your exam?
Overall nice, I guess.
Congrats. What was the lecture?
Thank you, hocam. Genetics 365.
From Emre Aksoy.
Yes.
Okay. So you should be tired. Your brain should be a bit tired, I guess.
A little bit tired, hocam.
Mm-hmm. Okay, guys. Let's make a poll. You're only 4 people, but I think we can make it. Okay, let's wait a little bit more. Muhammad Arslan is here. Hello, Muhammad.
Yes,
How is it going?
it's good. I have exam tomorrow, but it's pretty good.
Which exam?
Evolution. Uh,
From Mehmet Somer.
yeah,
Okay. Elif, how are you?
Fine hocam, you?
I'm fine, thank you. Okay. We're now a close group, quite close.
Yes.
It's perfectly okay. Normally the lecture should be like this. I think it will be better. Okay, let's share the screen. Okay, guys. This is the general scheme, what we were covering, just to remind you. And last week we were talking about mineral nutrition. You will have quiz, if you remember, from plant and cell structure, and also from mineral nutrition next Tuesday, and it will be face-to-face lecture. So if you remember, why minerals are important for us and for the plant, because this is the way plants get nutrition. Okay, they get the nutrition from the carbon dioxide, but also they get the nutrition from the soil. And some nutrients are needed a lot, called macronutrients, like nitrogen, sulfur, and so on. And some nutrients were needed in low quantities, iron, manganese, and so on. And what else we covered? Like in agriculture, the deficiencies of these nutrients are a big problem, and an optimum nutrition is very important. If you have families that grow plants, it's a very good idea to learn about nutrition and nutrition deficiencies, toxicities, and how to prevent them. I told you some techniques about how to prevent nutrition deficiencies. You have to first learn what they are and so on. So these were the topics we covered last week. And this week, in fact, still we are looking at nutrition, but from a much molecular place or subcellular place. We are gonna make a very focused lecture on how solutes, like nutrients, pass through the membranes. Why is it important? Because if we know how they pass through the membranes, we can modify the plants so that the good nutrients, let's say the nutrients humans need, can be directed to the edible parts of the plant, and the bad nutrients like cadmium, for example, or other toxic metals can be excluded from the edible parts. And you know, you can just make sinks. Sink means depot. By changing the nutrient concentration in one place, and then this will suck the nutrient from other places or deliver nutrients from other places to other places. What I try to say is, for example, if you make the root to suck more cadmium by just making its plasma membrane more permeable to cadmium and make it one-way transport into the vacuole, for example, then maybe all the cadmium in the plant, which is harmful for us, they will travel, because now there will be diffusion from high to low concentration. And since now you change the concentration of this heavy metal and create a sink, then you suck all of the heavy metal to the root, which is good for us because we don't eat the root, for example. I don't know if it was clear, but I mean, if we understand how the elements, nutrients pass through the membranes, we can do a lot. For example, we get our sugar from sugar beet, which is in the underground part of the plant. And if we understand how sugar is passing from plasma membranes, we can force more sugar to go to the root. So with the same effort, we can get more sugar. We can generate more sugar. So it's so important. We can manipulate transport, both cellular transport and the long distance transport, if we understand the membrane and membrane transporters. Okay, so these are the things we're gonna cover. By the way, if you have questions, comments, please tell immediately so we can look at those. So we are gonna work on solute transport from different perspectives. First perspective will be the energetic perspective. You all know from high school, like some transport processes are passive, some are active. It's largely dependent on the molecule. If the molecules are non-polar, then they can easily pass through the membrane. If they are polar, then they cannot. And we will examine this polar materials transport, including ions. So we will talk about water, which is itself polar. Then we will talk about the proteins on the membrane that facilitates transportation of these polar materials, called channel proteins. Then we will introduce a new term, which is cell voltage or membrane potential, which also determines the amount of this transport. And then we will later introduce the active transport, which further determines what will be transported, what will not be transported. And the active transport is divided into two: primary active transport. This is the transport processes that directly clears ATP. And secondary active transport, they don't use ATP, but they use the hydrogen concentration available in the cell. Okay, and then we move on to the kinetics little bit of transport, and I will introduce you high and low affinity transporters, because for the membrane transporters, they are not able to take whatever there is, but they are focused. They either are very active when there is little bit of material, and sometimes they are only active when there's a lot of this material. So we'll talk about that. And then we will compare channel and carrier type of transporters kinetics, which is quite different. And this information will help us to identify and characterize transport processes, whether the involved transporter is channel or a carrier. Then we will move on the how to identify and characterize new proteins on the plasma membrane, and I will let you know about the methods which we also use in our lab, like heterologous expression, heterologous expression expressing the gene in the yeast and look at it, patch clamp. This is a in vitro method. And then reverse genetic approaches are also possible using Arabidopsis mutant lines. So this is the outline of what we are gonna see. Let's move first to the energetics view or the transport processes. Okay, so this is chapter 6 already. Okay, I can pass that. Just a second. I will use the presenter view. Do you see now my notes?
Yes, hocam.
Yes, hocam.
You see the notes. Okay. Let's change it. Yes. Okay. I want you to understand and appreciate that just two molecules facing each other is enough to create a very different environment between the cell and the outside. There is nothing else. There is no concrete walls. There is no shield. There is no metal barriers and so on. Just two molecules, two phospholipids facing each other is enough to isolate largely cell from the outside. I think it's really amazing. And we have this also on the tonoplast, which is vacuolar membrane. So vacuole is also doing a lot of transport, and it creates sink. Sink means depot. Okay, this plasma membrane also is really great because it's the only part of the cell that contacts the environment. It has to sense whatever there is in the environment, and according to whatever there is, it needs to respond. And to do this response, it usually uses ions. So if there is a bug eating the cell, the bug's protein will be recognized, for example, and then it will trigger transporters of calcium, for example, which is a signaling molecule. And then this calcium levels change, and then this change then trigger gene expression and proteins against this bug. So it senses and it gives response using these metal transport from outside to inside and inside to outside. Okay, so this membrane lipid bilayer is not just made of the bilayer, but it has a lot of proteins on it. But so scientists try to understand how these nutrients and metals and whatever there is outside of the cell, how can they enter the cell, how can they go out? And if you need to understand something from the scratch, what should you do is you have to work on a very simple environment. So they started working on artificial lipids, which is made of just phospholipid bilayer, nothing else. And then they examine how the transport processes occur. What they found was interesting. If the molecule is uncharged and small enough, like oxygen gas, carbon dioxide gas, it can just diffuse through the membrane. So this is not a cell, this is an artificial membrane, just phospholipid bilayer. And the transportation was very easy. If the thing is charged, on the other hand, there was no transport. Doesn't matter how much molecules you have on one side, they never were transported to the other part. Okay, so if a molecule is uncharged, we call it nonpolar. If it is charged, we call it polar. And it's also possible that a molecule is uncharged but polar, which is the case of water. I will tell it in the next slide. And then we have the term hydrophilic. So for polar molecules, we call hydrophilic, loves water. And for nonpolars, we call hydrophobic, that hates water. Okay, and then we set diffusion. Diffusion is type of a passive transport, and we will later go on active transport, which is where the energy is needed. But as you see, for uncharged particles to enter into the other part, energy is not needed. So how is it possible for water not to be charged, but behave like it's charged, and what it means? Okay, water, I think, yeah, I have the structure here. It has this oxygen and hydrogens, and they are not like linear. So the amount of electrons on close to the oxygen is more than the hydrogen. I think you all know this. This creates a partial charge on the water molecules, and this changes a lot. So if we see the water on the leaves, you can see they are not everywhere, spread everywhere, but they hold like this. The reason they are like that is because every water molecule behaves like a magnet. So they attract each other, and in only this shape, the water droplet shape, the most attraction can occur. If they didn't attract each other, they would just lay down. So instead of a water, we drop oil, the oil will just go down because the oil molecules are not trying to hold themselves. So what is it similar to? I think it's similar to this. So you have the balls, but they are all kept together. They are not lying down because of this net. This is in water, the pull of each molecule, pull of molecules, and then every molecule pulls itself, and then it creates a net, like a net. Okay. So why is this important? Okay, but before moving to that, so what the scientists did was, when they have the lipid bilayer, the charged molecules couldn't pass. But if they introduce a protein on this lipid bilayer, which allows the charged molecules to pass, then we start to see the molecules can pass even they are charged. Okay. Yes, and this again was a passive transport. So what happens with the protein, and this is called channel protein, by the way, is it creates an environment inside which is hydrophilic charged, and outside a hydrophobic, so it can stay in the membrane, but then the hydrophilic part can let the hydrophilic molecules pass through. We will see its structure later on. We will see a lot of details and so on. So now I will try to give you the logic of how these things pass and why these things cannot pass when they cannot pass. And then I will ask you, okay? So please listen carefully and ask if you have questions. Okay, so I don't know if you see, if I point the right place, but the lipid bilayer, they have hydrophobic regions, and the uncharged particles, they are also hydrophobic. So what they do is they can move everywhere. Nothing is attracting them. Nothing is capturing them. So they are free. They just freely move anywhere. Okay. But for charged particles, they can't go to the other side. That's because charged particles are already captured by water molecules. They are covered with water molecules, which are also charged. And please look at my hand. So this molecule, in order to pass from the membrane, has to be released from the water because water will never want to enter the lipid. So because they capture the molecule, they cannot go to the other side. But what happens if there's a channel is inside the channel, you still have water. Therefore, it can directly go, or maybe it doesn't have water, but it has charged amino acids. So that these water molecules that are covering your molecule, they just stick to these charged particles. So your particle now, charged particle, is free to go because it's not captured by water anymore. So it can just go. Okay. So now I have questions for you. Here we have red polar molecules. And we have, sorry, this is wrong. I forgot to correct. We have red. No, it's correct. Okay, polar molecules. We have green polar molecules, and we have yellow non-polar molecules. We have a channel type transporter, and we have high concentration gradient, non-polar, polar. Okay, so we have high concentration gradient here and low concentration gradient at the bottom. Okay, so the questions are these 5 questions. I would like you to discuss about them. Normally, I was gonna send you to your rooms, but you can discuss right now. And I would like to hear. Yeah, please discuss them and answer the questions.
Red and green molecules cannot pass through the lipid bilayer just by themselves, so they should be hydrophilic, coated with water.
Mm-hmm. Do you agree, the others?
Yeah, I agree. It should be hydrophobic because it cannot pass through the lipid membrane, but
Hydrophilic, right? Hydrophilic. Yeah, so A is correct. Okay. And second question.
If they are polar, they should be charged, I
Hmm.
guess.
Yes. What about the others? Do you agree?
yes, of course, that is the reason that if they are charged,
Yeah.
they should be polar. That's why.
By definition, right?
It doesn't need to be charged to become polar, though. It
Also.
may be uncharged, but
Yeah, but it must have a charged region, right? I mean, in A, I said have charged region. I mean, like partially charged, you know, even water has a partial charged charge. Yeah,
yeah,
that
that's
okay, third one.
I think it must have uncharged region so that they can be attractive for charged polar molecules.
So A or B?
I think B.
B. Okay. What about the others? Say.
They should be charged, I guess, because there is there must be a channel for the pass, as I see here.
Yeah, you mean the oppositely charged to molecules, right?
Uh-huh.
For example, if I mean the inside of the channel should be just like oppositely charged to the molecules, the charge having molecules, so they could attract each other, and molecules can rush into the membrane because of that attraction between opposite charge. So I think yeah, you're right, they should be charged.
What do the others think?
As far as I know, the transmembrane proteins should have an hydrophobic region to become integrated into the lipid bilayer.
Yeah.
But having charge to attract charged molecules makes sense.
Okay. Yes, you were all correct. So they have a charged region inside, so that again the water covering the molecules, you should not see molecules just as molecules. They are covered with water. So these waters are released by these charged residues inside, and then it can pass through. I mean, inside is also charged, so these things you can consider them as like solubilizing inside. And as Ömer Faruk said, the outside of the protein must be uncharged because it interacts with the lipid bilayer. Otherwise, it wouldn't interact. It will be just sent away by the lipid bilayer. Okay, now the fourth question. How is it possible polar molecules do not pass through bilayer? So now I'm asking you a more like the logic of this. I already explained it, in fact, but I want to hear from you now. I mean, they are moving. Why do they move into the lipid bilayer? Why do they move only into the water? How do they know that they should not pass to the lipid bilayer?
Weak interactions with water.
So
Polar molecules are attracted by polar solvents, and because the lipid bilayer is non-polar, actually the water around the lipid bilayer causes it to have this shape. So inside of the bilayer is completely non-polar. Therefore, it repels the polar.
yes, so water doesn't let it go, right? Water doesn't let the molecule go into the lipid bilayer. Yeah, it's water that holds the polar molecules not to go to the lipid bilayer. Okay, fifth one. How is it possible lipid-soluble molecules pass to the other side? Why aren't they kept in the inside of the membrane? So you have these nonpolar things. They are now inside this nonpolar solution. Why don't they stay there?
They are all repelled to the same place, as far as I know.
They are all what?
They are all repelled to the same place by water, the lipid bilayer.
You mean, but so we have the lipid bilayer, we have the molecules, non-polar molecules, and they can solubilize in the this lipid bilayer inside, right? But then why do they go out? Because they are soluble here, but they are not soluble there. Aren't they attracted here? Because in previous condition, water didn't let the things go. But here, why do lipids let these guys go?
Maybe once they enter, maybe because of the concentration gradient.
Mhm.
Is it because of diffusion? Because small lipid molecules can diffuse
Yes.
directly through the lipid of cell membrane, and because of diffusion from one side to another side, they cannot be kept into the membrane and pass through it.
Okay, but the polar molecule, polar molecules, they don't go through the membrane because water was holding it. Although there is so the diffusion didn't work there. How does diffusion work for nonpolar molecules in the membrane? I don't know if it is clear. So we said they can diffuse from the membrane to the water. And now I'm asking, why aren't the hydrophilic molecules just diffuse into the membrane? Because they cannot pass the membrane. Although they are a lot in number, they just stay there.
Is it because of okay, I'm just guessing because of semi-permeability of membrane, that it allows some of them and do not allow.
Not really. I think because polar molecules are kept by the water molecules, so they cannot diffuse because they are not free. But nonpolar molecules, they are not kept by nonpolar molecules. They are still free. Because polar molecules are attracted to polar molecules, they are bound together. So the water molecules don't let these guys go to the lipid bilayer. But the nonpolar molecules, yeah, they are fine in nonpolar solvent, but there is no interaction between nonpolar molecules. So they can diffuse, they are still free. What do you think? Did anyone understand the logic? Okay, so first, polar molecules, they attract each other. So polar molecules are attracted by water. But non-polar molecules, they don't attract each other. Is it okay?
Yeah.
Yeah, they don't attract each other because there is no charge that keeps them together. This then creates the difference. Because now polar molecules in this part of the bilayer, they are hold there tightly. But non-polar molecules, they are not hold, they can come to the membrane. Since they are also not held by the membrane, they can diffuse to the other place, the bottom.
But hocam, if they are not wanted by the lipid bilayer's inner side, how can they enter the membrane in the first place?
Because I think since they are not attracting any part and they are not repelling anything, so then there is no repel force on nonpolar molecules. There is no attraction on nonpolar molecules. Just imagine you have the nonpolar molecules. They are just going like this, and if there are many, some of them will hit the I mean, they already hit the non-polar part, okay? And inside there is polar part, and then they hit through the polar part, and polar part doesn't care that much. So they just find their way because there is too many of them. By diffusing, they just diffuse inside, and then they diffuse to the other side also. Since there is no one capturing them.
I think I get it.
Okay. So the others.
Just clear hocam.
Okay. Okay. Yeah, just think about it. If you get the logic, then you can solve many problems. Usually people make us memorize things, but I think this is understanding that is very important, really. Then later on, you understand many different things. Okay. Okay. So this is lipid bilayer again. We have a video. I will show you. Just a second. Yes. What I want to show you is these charged particles, when they enter inside, they create a charge difference between two parts, in and out. And this charge difference then determines the transport. So scientists saw that, for example, let's say you have 10 minus charged particle. If you wait enough, then you will have 5 minus charged particle and here and 5 minus charged particle below. But if you have another material and if it is like +2 charged, then due to the charge differences, sometimes the number of the molecules in two sides of the membrane is not equal. Scientists saw that. So I want you to watch this video. Let me play it, and then you will understand what I'm gonna say, because it's, I think, tells very nicely. So what I try to say is, it's not only the so by diffusion, normally we think every molecule in two parts should be equal. But now we have to think about the charges also. When we think about the charges, now the number of molecules don't have to be equal in both sides. Okay.
Membrane potential or membrane voltage refers to the difference of.
Can you hear easily?
Yes, hocam.
Okay.
Yes.
Membrane potential or membrane voltage refers to the difference of electric charges across a cell membrane. Most cells have a negative transmembrane potential. Because membrane potential is defined relative to the exterior of the cell, the negative sign means the cell has more negative charges on the inside.
Hocam,
Two basic rules.
Yes.
do we need to see something or are we just listening?
Yes. You have to see the video.
I can't see the video, hocam. It's
Okay,
just me.
sorry. Okay. Now you will see it.
Yes, hocam, now I can see.
Yes.
Okay, we are trying to understand what will happen if we put ions. How many of them will pass to the other side, okay?
Membrane potential or membrane voltage refers to the difference of electric charges across a cell membrane. Most cells have a negative transmembrane potential. Because membrane potential is defined relative to the exterior of the cell, the negative sign means the cell has more negative charges on the inside. There are two basic rules governing the movement of ions. They move from higher to lower concentration, just like any other molecules. Being charge-bearing particles, ions also move away from like charges and toward opposite charges. In the case of the cell membrane, there is a third factor that controls ion movement: the permeability of the membrane to different ions. Permeability is achieved by opening or closing passageways for specific ions, called ion channels. Permeability can change when the cell adopts a different physiological state. Consider this example. Two solutions of different concentrations of sodium chloride are separated by a membrane. If the membrane is equally permeable to both sodium and chloride, both ions will diffuse from higher to lower concentration, and the two solutions will eventually have the same concentration. Note that the electric charges remain the same on both sides and membrane potential is 0. Now let's assume that the membrane is permeable only to the positively charged sodium ions, letting them flow down the concentration gradient while blocking the negatively charged chloride ions from crossing to the other side. This would result in one solution becoming increasingly positive and the other increasingly negative. Since opposite charges attract and like charges repel, positive sodium ions are now under influence of two forces. Diffusion force drives them in one direction, while electrostatic force drives them in the opposite direction. The equilibrium is reached when these two forces completely counteract, at which point the net movement of sodium is 0. Note that there is now a difference of electric charge across the membrane. There is also a concentration gradient of sodium. The two gradients are driving sodium in opposite directions with the exact same force. The voltage established at this point is called the equilibrium potential for sodium. It's the voltage required to maintain this particular concentration gradient and can be calculated as a function thereof. A typical resting neuron maintains unequal distributions of different ions across the cell membrane. These gradients are used to calculate their equilibrium potentials. The positive and negative signs represent the direction of membrane potential. Because sodium gradient is directed into the cell, its equilibrium potential must be positive to drive sodium out. Potassium has the reverse concentration gradient, hence negative equilibrium potential. Chloride has the same inward concentration direction as sodium, but because it's a negative charge, it requires a negative environment inside the cell to push it out. The resting membrane potential of a neuron is about negative 70 millivolts. Notice that only chloride has the equilibrium potential near this value. This means chloride is in equilibrium in resting neurons, while sodium and potassium are not. This is because there is an active transport to keep sodium and potassium out of equilibrium. This is carried out by the sodium-potassium pump, which constantly brings potassium in and pumps sodium out of the cell. The resulting resting potential, while costly to maintain, is essential to generation of action potentials when the cell is stimulated.
Okay. So guys, I don't know if you get it. What cell does is it put channels for some ions and not for some other ions.
Vraiment,
Sorry.
c'est la seule chose qui compte pour moi. Ma seule raison de vivre.
Okay. Yeah, it just puts some transporters for some ions and they put less transporters for some others. So they create a difference in number of ions that passes. By just doing that, they create a membrane potential. They become like a battery. They just let some ions pass easier, some ions pass less easier. By just doing that, they create like a battery. a voltage. And this voltage of the cell also determines how many ions will pass there and how many ions will go back. It's a bit complicated to understand, but I hope you get it. You can ask. Yeah. And you can watch the video again and again. Okay. So now I will start share the screen again. Okay. Although here sodium concentrations are different in two parts, the electric charges, I mean, there's a equilibrium, although the number of sodiums are different. Even if you let it stay in 100 years, there will be no diffusion. Normally, we thought after diffusion, the number of elements equilibrate, but if there is this charge and selectivity in the membrane, then it is not. We need to understand that, because then only then you can understand how ions, I mean, which ion will enter the cell, which ion will not enter the cell. You can understand that only if you understand here. Okay, then what scientists did after working on the membrane, the artificial membrane, an easy way to work, then they said, okay, now we know how the ions are transported into and out of the membrane. So now if we measure the voltage between the cell and the solution and the concentrations of ions, then they thought they can predict from the voltage how many atoms should be inside the solution and inside the cell. And then they measured these solutions, and then they checked if the predicted value fits to the actual value. Okay, what they did is they put actual cells into the solution, and then they measure the voltage. Let's say minus 70 volts, okay? And then they put a known number of element here, like known number of potassium, sodium, calcium, and then they predicted if all diffusion, or if all this process is happening as if it is happening in the artificial membranes, there should be 74 potassium, for example. And then when they measured the value inside the cell, they measured 75. So it was exactly how they predicted. But when it comes to sodium, they saw that the prediction was 74, but the observed was only 8. So observation was different than the prediction. Later on, we understand why this inaccuracy happened, because the cell can do active transport. So if it was just passive transport, it would be as the predicted values. And you can immediately say for potassium, the potassium transport is just passive transport. But sodium transport is active, meaning the cell, by spending energy, took sodium inside. And for example, for nitrate, it's the vice versa. Sorry, I correct myself. For sodium, it pumps the sodium atoms out of the cell. And then for nitrate, it took the nitrate ions into the cell. So there is an active transport. Okay. Just let me share again. Yes. Okay, by the way, this was just to tell you or introduce you the concept of active transport. So for passive transport, we can predict what we will observe. by considering the concentration and voltage, but when there is active transport, it's very difficult to observe, predict. By the way, these cells are over simplified. So normally inside the cell, you have compartmentation of the elements, and yeah, but just for example, we showed these numbers. Okay, so what the scientists did to examine the effect of this energy-driven transport, how effective it is, this active transport, they put a chemical called cyanide, which blocks the active transport, and then they measure the voltage, the membrane potential, and what they saw was normally the membrane potential was 130 millivolts, but when the energy is taken out, so now if it was like artificial membrane, then the voltage would drop to -50. Okay, so most of this voltage difference, I mean, until -50, the voltage difference was due to passive transport, because some ions were let inside, and some ions let inside in different rates or let outside different rates. But then the rest of the potential was due to the active transport of ions. Okay, and then I will also tell another mechanism how the cell mediates the membrane potential, which is by directly sending protons outside by pumps, spending ATP, but we will come to that. Now I have one question for you. Okay, this is the question. Please answer these 3 questions by observing the graph. First question is, which of the ions and compounds have transporters in the biological membrane? I give you enough time, just read the x-axis, y-axis, try to understand what's going on, and then start discussion. By the way, will you feel more relaxed if I put you in a room and then I won't hear, and then you discuss and then you come up with suggestions? Would you prefer that?
It is okay, I think, hocam.
Okay.
So in the first question, we need to check that whether they have transporters or not, right? I mean, I know that the ions, there are metal ions like sodium, potassium. They
Yes.
need to be transferred through transporters as compared to other ions.
Okay, first yes, then I will come. Yeah. Please rephrase your question and then your colleagues should answer.
Son soruyu anladınız mı? Neden bahsettiğini anlamadım ben.
Mohammed cannot talk English. Sorry, Turkish.
Yes, hocam, I cannot.
Yeah. So if you can speak in English, it will be better.
Okay, I'm sorry.
Yes, can you tell your opinion to others and others evaluate whether they think it's correct? This is the discussion I'm expecting.
Yeah, I think that the ions, the metallic ions, they need transporter to pass through the membrane because they cannot pass by themselves. So they need active transport, some protein to transfer them through the membrane as compared to the other like water and glycerol.
Okay, maybe I should give the information that the lipid bilayer itself doesn't have any transporters. Okay, does a biological membrane transport sodium more or the lipid bilayer?
Could you please just give the hint? What do you mean by biological membrane? I mean,
Yeah, biological membrane is from the cell. They kill the cell and take the lipid bilayer, but it has proteins on it. This is the biological membrane.
okay. I mean the most outer layer, right?
Yes, the most outer layer. So it has proteins. It's lipid bilayer with proteins, and the other lipid bilayer, the artificial lipid bilayer, is a just lipid bilayer. Yeah, that's it.
So I think efficient to transport the ions like sodium and potassium as compared to the membrane having transporters as proteins.
Why do you think like that?
Because these ions require some carriers or transporters to pass through the lipid bilayer membrane. They cannot pass by themselves. They need active transport, which is done by the proteins. So the lipid bilayers provide that protein to transport these specific lines.
Okay. This lipid bilayer, the artificial one, is also permeable because they use a technology and they make it permeable to potassium, sodium chloride, little bit at least. Okay, so again, maybe I should ask like potassium, is it more permeable in the biological membrane or the lipid bilayer? Is potassium more permeable for biological membrane or in the lipid bilayer? In chat, I see an answer. Yes, biological membrane. That's correct because the permeability is 10 to the power of -6, and in lipid bilayer 10 to the power of -10. This is extremely low, 10 to the power of -10. Therefore, yes, the biological membrane permeates potassium more. And in fact, all the things top of this line says biological membrane can permeate them more. Therefore, we can say for all these things, there must be some transporters specific to those things in the biological membrane. Questions about that or comments?
So the molecules like that are non-polar, like oxygen and carbon dioxide, they are already, soluble to lipid bilayer, so they don't need any.
Yes and no. So for non-polar things, like oxygen and carbon dioxide, still you have a permeability issue. So yes, there will be diffusion, but it might be very fast or it might be very slow, depending on the membrane structure and the transporters. If there are transporters, then they will be transported much faster. Or the size of the molecule, if they are small, like oxygen, I think is smaller than carbon dioxide, so it's more permeable. Okay. So then second question is, why do glycerol, carbon dioxide, and oxygen have different permeability? Are there different transporters transporting them?
It can be about size, I think.
Yes, probably it's not about transporters because
Mhm.
in both lipid bilayer and biological membrane, the transport is the same, which means it's probably not about the transporter because we have the transporters in biological membrane, but not in lipid bilayer. But still they have the same speed, but they are different from each other. Therefore, it should be maybe about the charge, although they are uncharged. I think there is nothing completely uncharged. It should have a little bit charge, and they should have a size and so on. Size differences. Okay, the third question is more difficult to understand. Would it be theoretically possible to have an ion or compound below the line? Below the red line, I mean. First, what would it mean, and would it be possible? I don't think so, Ömer says. What do the others think? Okay, if there is a compound below the line, it would mean that with a membrane which has transporters is less permeable. But how can it be possible? I mean, if you have transporter, then it can either be more permeable or equally permeable. Because maybe the transporter transports something else, but not that one. But it's not possible to have less permeable membrane with transporters, I think. So transport, if you have transporter, either the permeability would increase or it would be the same. It's not possible to have it opposite way, I think. Yes, and the cell is dead. Yeah, Ömer Faruk says, yeah, and the cell is dead. There is no active transport. Yeah, there is no active transport here. Okay. Let me correct a little bit. No active transport, I will say. No active transport. Okay, let's give a 10 minutes break. So it's 17:40 now. Sorry, in my time. So 50 past, we will continue. Hello, guys. We have just one more minute. Umut is also with us. Merhaba Umut.
Hello hocam. I am sorry. Today I had another lecture
Of course.
until 8 p.m.
Yeah.
So I came a little late.
Yeah.
Not more than a little late, I'm sorry.
Yeah, because this is an unusual lecture. I don't expect all people to come. So that's perfectly okay. Thanks for coming.
You're welcome hocam. Thanks for giving us this lecture.
Okay, guys. We can continue, I think. Just a second. Yes, we had this difficult problem. Okay, with this, we come to the proteins. Now we will see what kind of proteins are there on the membrane. And we should say, so this is the lipid bilayer and these are the proteins. There are many different types, but we can group all of them into three. So one of them, I mean, as you can see here, we have the channel proteins. So they are involved in only simple diffusion. So they just open a gate and then things pass through it. So maybe a question to you all. I mean, if there is an open gate, like hydrophilic region, why don't everything pass through there? How can it maintain the selectivity?
Uh,
Yeah.
Maybe by size hocam. Adjusting the size of the channel, we can prevent larger molecules to enter or get out from that particular channel.
Okay, good. Other ideas? Other ideas?
If it is active
Mhm.
transport, we need some proteins, transporter proteins.
Yes. But this is just simple diffusion. So the question is like, you have molecule A, which is a lot, and then it passes through this channel protein, and channel proteins are like open doors. So why not everything enters from the door once it is open? So just imagine if you left your house door open, animals will enter in. So how does the cell still makes the selectivity? Okay, I think a good analogy to that is this. I don't know if you can easily see. So the doors have shapes that is only possible for certain shape molecules to pass. Can you see easily the picture?
Yes hocam.
Yeah. Okay. So this is how channel proteins work. And then we have the active transport. Sorry. Yeah, we call it again passive transport because there is no ATP spent. But yes, still it requires some proton gradient. I will tell in more details. These are carrier proteins. Carrier proteins transport things, but it's a different way of transport than the channel proteins. So this thing binds to the carrier protein, and then this binding changes the conformation of the protein. And now the protein changed the conformation and the solute passed through the other side. Compared to the channel proteins, they are very slow because there needs to be like real interaction. But in channel proteins, it's like open door. Okay. Okay, so carrier proteins are also passive. They don't require to spend energy, ATP, but they can passive transfer. Yes, they can transport against the gradient. It's possible. You may have a lot of element A here, and then you can still transport element A there if you have a gradient. I will tell in more details. So without expanding ATP energy, you can still do this with carrier proteins. And the other type of protein is also called pump. It uses ATP. We usually call them ATPases, or pumps. There are many types like calcium pumps, zinc pumps, and so on. They use energy to send molecules against the gradient. Okay, then for carrier proteins, I have another analogy, but I will tell it when we come to that. So, but you remember this scene from the Turkish movie, I guess. Okay, we will come to that. Okay, before telling more about the transporters, I want you to understand how this open gate works, or even the other transporters, they have similar logic. So a transporter, membrane transporter, has two parts, hydrophilic and hydrophobic part. The hydrophobic part is integrated in the membrane, lipid membrane. At least the part of the protein that interacts or faces the lipid membrane are non-charged, let's say. Okay, and then maybe they dimerize, trimer, or I mean, these proteins, the same protein, bind each other many times. It's like that, and then they make a hollow structure, and inside is empty. And this is where the nutrients can pass through, or the elements can pass through. So you should think the picture in B in 3D. So a protein has many transmembrane domains, but you should think this as it's making a hollow structure. Okay, and what is hydrophilic parts inside and outside doing? They are important usually for recognizing the molecule, or like they have some other functions. Maybe they get phosphorylated and then they open the pore size more or less, and so on. They turn on or turn off. So the regulation purposes, they can work for regulation. Okay, again, these proteins create a hydrophilic region inside and hydrophobic region outside, which is anchored in the membrane. So for scientists, it's very important to understand what region is important for what reason for these proteins, because then they can manipulate the proteins. Many of those proteins, they are specific, but sometimes they show less specificity. So for example, a potassium carrier, if there is a lot of magnesium around, maybe it also carries some magnesium, and maybe you don't want this. Then what you can do is you get the sequence of this protein, put it in yeast, and then do mutations, and then see if it still carries potassium, but much less magnesium. So you can search for a protein which carries your your metabolic of interest more, and non-specific things less. A very good example to this is iron transporters. There is an iron transporter called IRT1. It also transports cadmium. So when plants need iron, plants also accumulate a lot of cadmium, which is very bad for the health. So what the scientists are trying to do is they take the sequence of this gene, and then they do mutations and try to find a protein which can only take iron but not cadmium. Okay. Or what they can do is maybe they can change these regulatory parts, so they make the pore bigger, or the number of pores can be increased, and so on. Okay.
Hocam,
yes.
can I ask a question?
Sure.
Are the drought-resistant plants also made by this method too? Like we know why the plants become yellow and dry when there's a drought. So can you prevent this with this method too? Like increasing the carbon dioxide specificity of the protein?
Yeah, so I think drought is related to many things, but mostly related to water transporters. We learned in the last 10 years, I guess, that water needs to be transported by transporters. And these transporters are called aquaporin family. So if we modify these aquaporin proteins, then we can make more drought-resistant plants. One way to do this, maybe we can find already water-resistant plants, and then we can search for what gives them this resistance, and then we will come up with some maybe water transporters, and then we can just mimic these water transporters or take them and put to the our plant of interest. So it's very relevant question, and yeah, you're right.
Thank you hocam.
Okay. Okay, so what about the pumps? Now I will tell you about these ATP requiring pumps. Okay, to me it's similar to this movie. So I don't know if you know this very famous Turkish movie. Münir Özkul, the main guy, he earns the money, and then he gives the money every week to every guy. So in this big family, he is the only guy who earns the money. I think he's similar to the pump. So he works and he earns the money. So the pump spends ATP, and then it creates hydrogen, proton. We call it proton motive force. So he creates this charge difference, and all the rest of the family or the secondary transporters, they use this charge difference in order to do things. So this is the analogy I come up with. If the guy who doesn't earn anymore, all the rest of the secondary guys, they cannot spend any money. And as a transporter, if the pumps are not working, ATPases are not working, then there will be no voltage difference between two sides. And all the secondary transporters that work with this voltage difference, they don't spend any ATP themselves, they will stop working. Okay, so what are these secondary transporters doing? So the other guys in this picture. What is secondary transport is they do use energy, but this is already accumulated energy as hydrogen. So here in the picture, you can see if there's a lot of hydrogen on one side, the transporter can use it, and then it can send elements against the gradient. In the picture, you can see how this is done. So hydrogen enters the empty place, and then this changes the conformation. And now this conformation change is so strong that it can transport the nutrient or the element even against the gradient. This is one way. There is also another way. These transporters can be two types. They are either called antiports or symports, depending on how they do the transport. So in the left picture, you can see in the symport, hydrogen and the elements are transported in the same way, and in the antiport, they are transported in opposite directions. And to analogy to those, I think for antiport is this, what is it called? Does anyone know? Tahterevalli in Turkish. So you have hydrogen, it goes this way, and the element goes the opposite direction. But if you are throwing the arrow, it's I think similar to symport, because both the sting, is it called sting? Rubber, let's say, and the arrow, they both go together. And the arrow goes without spending any money or any energy other than, yeah, the rubber will do this. And rubber has this potential accumulated, and then it will just use to send the arrow. Any questions? Okay, so there's no real ATP exhaustion here. Okay, and then I have one slide where I want to tell you the logic of any transporter. So basically, how does this thing work? You know, you have hydrogen, and then there's this change, and then, I mean, how does it work? I just want to tell you like how I see it. So one of the loops, let's assume, it's very specific to one metal. Let's say hydrogen. Iron, let's say. How can it be very specific? Because some amino acids, they like to bind to iron, and iron has a structure, and the amino acids, if they really localize perfectly, maybe this atom, iron, can bind there very easy, and then now you have a very specific structure. So when this thing binds there, now the it's there stable. And let's assume for antiporter, inside the cell you have a structure which can bind to hydrogen. So when hydrogen binds to this, then there will be a structural change. So now what happens is, maybe just an imagination, this iron there, which is captured, maybe another region of the protein comes, and then it binds to some of the amino acids that were previously bound to iron. Now, because the iron binding amino acids are now binding something else, okay, now iron is released, but maybe because of the conformational change, now there is another protein part which blocks the way. So iron is now released, but it cannot go that way, back to the side water. So the only way it goes is now to the cytoplasm. So what I try to say, you have first sensing and strong binding, and in other parts, when the hydrogen binds, the conformational change occurs. Some proteins come bind with the things that normally loves and binds iron. Now iron is free, but it cannot go away because now the gate is closed. Does it make sense? So if you imagine like this, I think it's very somut. It's not abstract anymore. So this is the logic of every kind of transport you can imagine like this. I mean, of course, there are many differences, but this is one way to imagine. Okay, with this, I told you about the transporters, the kinetic, the energetic view. So my focus was on like which transport requires energy, what type of energy, and so on. But there's also a kinetic point of view. So these two graphs, although it's just one slide, it's quite dense, so you have to listen carefully. If we have a simple diffusion, then as the external concentration of transported molecule increases, you will have steady increase in the cell of this element. But if it is transported by a carrier protein instead of a channel, so in first case we have a channel. If we have a channel, there is a passive diffusion, and then as the element concentration increases, there will be steady increase in the diffusion. But if there is a carrier transporter, then since it works like an enzyme because it has to bind, and if you increase the concentration of the chemical, then all the sites would be bound already. So it will be saturated, just like an enzyme. So it will follow an enzyme kinetics. as you increase the concentration, there will be not a linear increase in transport, but it will go like this. It will follow the Michael-Menten kinetics. By just increasing the concentration of something in the solution and looking at this kinetics, you can immediately say what kind of transport is there. Is it a diffusion? Is it channel, or is it a carrier protein? You can immediately say that. So this figure 6.12 is an example. So please look at this and try to tell me what is there. Please read it yourself. What is strange there is you have one line and then you have another line. So what's happening there?
So, hocam, aren't these graphics are what happens when there is no enzymes or carrier proteins, and what we observe when they are at the area? Like
This is
on
an
the
actual graphic, actual cell. You increase the sucrose level in the medium because you want to understand whether sucrose is transported passively or actively, okay? This is what you are trying to do. And this is the actual result you get. You measure the sucrose concentrations by time and by, external concentration, and you come up with this graph. It's very strange. Okay, I will tell. So what happens is until 10 millimolar sucrose, you see an enzyme kinetics, which means there is a transporter, there's an active transporter. But then it's saturated. And as soon as it's saturated, you start to see there is also a passive transport of sucrose. But you can only see this after your carrier transport is saturated. And this is, in fact, this reflects very nicely what is happening on the cell membrane. Usually you have many transporters for the same thing. Some work very well when the concentration of the material is low, and some work much more when the concentration of the material is a lot. So if a transporter works in low concentration, we call it high affinity transporter. If the transporter works when the elemental concentration is high, then we call it low affinity transporter. It's extremely important in my area of research to have low affinity transporter or high affinity transporter or both. So here, the first transporter, the carrier transporter of sucrose, is a high affinity transporter because it operates when the sucrose level is low. So just imagine you are a cell and you have very little sucrose, you have to get it. So you put a transporter which really takes whatever there is, okay, against the concentration gradient. It takes every bit of sucrose from outside to inside. So the first transporter is high affinity transporter. And then if you have a lot of sucrose outside, then the cell doesn't care anymore. This high affinity transporter is not needed. You just open the way and then more sucrose comes in. So I guess the cell sucrose level should be maybe like 7, 5, 8, something like that, but not more than 10, because more than 10 you have a passive transport, which means outside sucrose concentration is now higher than the inside sucrose concentration. So this is so logical. If you didn't get it, if you get bored, that's okay, but go back again and then try to understand, make sense of this. Any questions here? Okay. Okay, guys, with this, I finished the kinetic point of view, and now I will tell you how people identify and characterize transporters at the first place. So to understand this, there are terms to learn, and then I will tell them one by one. But you need to listen carefully and maybe take notes. You will find them in your book as well, but maybe not as detailed as I explain. So in your book, these things are under the topic, the genes for many transporters have been identified. Okay, so first, the terms identify and characterize. These are two different things. Any idea what makes them different? What is different identifying a gene and characterizing a gene or a transporter?
Characterizing, I think, is specifying the gene for something specific character or quality, and identifying is just to identify, I mean, just to discover or check the normal of basic concept of that.
Very basic information, yeah. So exactly, identifying is like this sequence transports zinc, for example. This is identification. And you name the gene, I call it WIT1, and then this transports iron. This is identification. In the genome, you identify a certain region of genome as a transporter protein. And characterization means under this circumstance, this transporter is very active, and then without it, maybe the flowers are dying and the plants will die. Okay, so in every different condition, you may have a different characterization. So characterization is almost endless. Once you identify the gene, you characterize it in 4 different conditions, and then try to assign a physiological function for the gene. Okay, second term is Arabidopsis tDNA insertion mutants. So people use it to identify genes, and they use it in reverse genetic screenings. What it is you have the plants that are mutants for just 1 or 2 genes, and then you put these mutant genes in a condition that is normally just a little bit challenging for the wild type plant. And then now if the mutant is challenged much more and showing phenotype, then you say the gene it lacks is involved in tolerance to that condition. For example, you are searching for genes, transporters that transport sodium. What you do is you get these tDNA insertion mutants and put them in salt-containing medium. And normally wild-type plant performs still okay. So this salt level is not detrimental. But if some of the mutants are dying, then this means the genes that are mutated are required for this condition. So this is called reverse screening, finding genes, identifying genes with reverse screening. You just buy these tDNA insertional mutants from internet. They are sold. There are many. There are maybe 30,000 mutants for each gene, and this is how we identify and characterize genes. This is one way. Okay, this is called reverse screening. Another way how we examine the transporter proteins is by complementation. So what we do is we use yeast. So here in the picture A, you can see the yeast cells. so these transporters, if you express them in the yeast, then they also work. And for yeast, we know a lot of mutants that are unable to do certain things. Like maybe there is a potassium transporter lacking yeast mutant, and you get it, and then you prepare, and then you put your plants under potassium deficiency, expecting potassium-related transporters are now more expressed. And then you take the cDNA you make a cDNA library. I don't know if you know these terms, but you should, I think. And then you express the cDNA library in the yeast, and then whatever cDNA now gives your yeast tolerance, then you say, this cDNA or this gene is a metal transporter. So this is called complementation of a yeast mutant. Or you don't need to do complementation, you just get the wild types yeast and then one by one express all the plant genes on this yeast and then see now whether they transport more metals. Okay, this is another way how to characterize and identify metal transporters or other transporters. And there's another method called Xenopus oocyte. This is on the picture here on the plate. So this is just one cell, but this cell is very big. It's a cell from the frog. It's the egg cell of the oocyte cell of the frog. It's very big. What you do is you put your gene in it, and then you examine the concentrations of the chemical outside and inside of the egg cell. The advantage it gives is normally cells are extremely small. You cannot put electrodes, but since this is very big, you can put electrodes and then you can measure the difference of this chemical, the concentration difference. If this chemical is now too much, a lot in your egg cell, then you say the transporter is transporting this chemical into the egg cell. This is how you characterize. And then there's another method called patch clamp. Patch clamp is also a very great method. So how this works is you go with your micropipette and you dip it into the cell, and then you just push back, and what happens is your micropipette tip will have the membrane. So you will have a membrane here. Inside you have liquid and outside you have liquid. And then you measure the difference between inside and outside. This way you can identify the transporter in this membrane. Or I mean what it is transporting and how it is transporting. You can do your research like that. And we have another term called heterologous expression. Heterologous means expressing a gene in a different organism than its original organism. So if you express a plant gene in frog or in yeast, then it's called heterologous expression. Why do we do that? Because the protein in the plant is interacting with a lot of other things, but we want to put it in a very simple environment, like yeast is much more simpler, just one cell, and then or egg cell. So in very simple environment, whatever difference you see is a direct, you suppose it's a direct, it reflects the direct property of the cell. Sorry, transporter. Okay, and then we have the term phenotype. This means when you express something in the wild type and it behaves different than the wild type, we say it shows a phenotype. It's any difference that's deviating from the wild type. Okay, so how is it now time-wise? Let me see. It's 6:30 to my time. We started 4:30, so it's already 2 hours then. And the last method we can do is like looking at homologies. If a transporter is found in another organism, you can just examine it and compare it with the organism of interest, and then you can make some deductions like this transporter transports manganese is another another organism. In my organism, it should also transport something similar to manganese, for example. Okay. Okay, I have one question to ask, and then I will let you leave after making a short summary. Okay, let's ask this question. Okay, this is a quite difficult question. So these are your answers. The correct answer should be, I think, D, A and C together. I told you about this homologies. Whenever you see a transporter in humans, if it is identified, you immediately should go check with the plant ones. And B is not correct because, I mean, if you already know the protein and then synthesize it and then compare now the newly expressed membrane and compare two membranes where your protein is expressed and where not expressed, you can get some ideas, but without prior knowledge of the gene, you cannot do that. Okay, any questions? Okay, guys, I want to thank you because you were quite active. I know it's very late and you were tired. You had exams and you have exams, but you still try to focus on what we are talking about. And although these are really dense things, so I just want to tell that I appreciate that very much. I'm very thankful to you for coming. If you have any questions, any comments, please let me know. And you can tell if you have suggestions always by pushing the button in the ODTÜ class, like if something was not clear to you, or yeah, just let me know. Okay. So take care. If you have anything to say, just say. Yeah.
Thank you for the lecture, hocam.
Okay.
Thank you for
I-
the lecture.
Can I ask something, hocam?
Sure.
Since I don't have this course officially, I can't reach the class and I can't reach the presentations.
Oh,
Could you
really?
add me, please?
I will add you immediately.
Okay, hocam, thank you so
Okay.
much.
And on Tuesday we have the quizzes, guys. So please study for those, and let me know if you have any questions.
We have one quiz, hocam, right?
Don't you have two quizzes?
In ODTÜ class, there is different dates, so
Okay.
I wanted to ask.
Okay. I will double
We
check.
have two?
If it is like that, probably you have two quizzes in different dates. You may be right, like Tuesday
Okay.
and Friday. I will double check. Okay.
Okay,
Thank you.
hocam.
Take care.
See you.
And next lecture, we will see each other because I will be back. Okay, bye bye.